Hey Team Bio, welcome to your last screencast on the Big Four, Part 4, DNA and RNA. Ooh, way too big. DNA and RNA. Okay, together DNA and RNA are both composed, or they are nucleic acids. How do I want to draw this? DNA and RNA are nucleic acids. And they have two kind of discrete functions in cells. The function of DNA is it encodes genetic information. And the function of RNA is it's kind of short-term information. Oops. Storage. As well as sometimes RNA can modify the expression of genes, so gene expression. And it can also sometimes be an enzyme. So you probably heard a lot more about DNA, but RNA has a much more kind of active role in regulating how DNA is expressed. And I hope you remember early on in the year, we talked about the central dogma of biology. And it's this idea that information flows from the DNA to the RNA and then to proteins, which lead to the traits and behaviors that we see that are genetically encoded. So DNA is like the long-term storage molecule, and then RNA is kind of the action molecule. More on that kind of later in the year. We'll talk about this whole process of transcription, the process of taking DNA and synthesizing RNA off of it, and then the process of translation, taking RNA and making proteins from it. So really early on in the year, we looked at this image and labeled it together. And what I want you to do is pause the video right now and do your best to label it all over again without any notes. So pause right now, label it again, and then when you're ready, pick back up and we'll see how much you can get. Okay, so I'm just going to go ahead and label it right now. We saw that this was a cell here, and you know that this is the nucleus. It contains the DNA. And there's also this structure inside of it called the nucleolus, which is the site of ribosome synthesis. More on that later. There are also some organelles in here. We know that this is the endoplasmic reticulum, or the ER. This is called the Golgi body. Here are mitochondria. And we know that this whole thing is a cell. Right away, looking at the cell, I can tell a couple different things about it. Number one, I know it's a eukaryotic cell because it has a nucleus and membrane-bound organelles. And I also know that it's an animal cell because it doesn't have a cell wall. If it were a fungal cell or a plant cell, it would contain a cell wall, and it doesn't, so I know it's either a protist or an animal. Okay, so within the nucleus are coils and coils of DNA. They're called chromosomes. So chromosomes are DNA coiled and then once again supercoiled on top of those coils into these tight structures that in different phases of the cell cycle can either be tightly wound or loose. But the whole structure, I guess I should say that, what do I want to say? The DNA is really very specifically organized and packaged around proteins called histones. Oops, there's an E in there. Histone is a protein. And the DNA is wrapped around these histones, and the DNA and histone complex is called a nucleosome. So we see the DNA here, kind of in this vantage point, and the DNA here. So these are both pointing to DNA. Remember we said that this was a gene, which is a location on a chromosome. And this gene could be the gene for, let's say, hemoglobin. And different folks have different sequences for that gene. And so the sequence of nucleotides at this gene would be the allele. Because we are all humans, we share all the same genes, but what makes us different from one another are the alleles that we have at those genes. Did I spell this right? Allele, A-L-L-E-L, yeah, E, yeah. And, okay, and then, well, what is a nucleotide? A nucleotide is the most basic unit of DNA. So this box represents a nucleotide. And there are 4 different types of DNA nucleotides. We have cytosine, guanine, adenine, and thymine. We often just abbreviate these as C, G, A, and T. I think that was most of what we labeled. Oh, and then we also said that DNA has a sugar, phosphate, backbone, which was here and here. And actually I think it's helpful to draw this out as well, which might be a little annoying, but hopefully, I promise it'll be worth it. Okay, so our monomer. We're back to discrete, excellent monomers here. Our monomer of nucleic acid is both a DNA nucleotide and an RNA nucleotide. DNA and RNA are very structurally similar to one another, except DNA stands for deoxyribonucleic acid, and RNA just stands for ribonucleic acid. There is one oxygen group, or it might be a hydroxyl actually. Let me look that up. DNA versus RNA structure. A sugar ribose, deoxy sugar, yeah, okay. So it's a type of ribose that lacks one oxygen atom. Oh, my original instinct was right. Okay, so structurally they're very similar with a few key differences. A DNA nucleotide is made up of a 5-carbon sugar. So we'll draw that out here. Okay, excellent. And then it has bonded on this side a phosphate group, and I'm going to draw this as well. You'll see in a second. And then over here, it has a side that is composed of a nitrogen base. So this is a sugar group. Here's a phosphate group. And this is a nitrogen base. The flavors of DNA, as we said before, come in A, T, C, and G. And just for continuity, I'm going to color code them like they were in the picture above. So C is in red, G is in teal, A is in yellow. Oh, I don't want that yellow. Oop. And thymine is purple. Okay, so these are our 4 different flavors of DNA. Now, RNA has a very similar structure overall, but I said before it's missing, DNA is missing an oxygen at one point off of the sugar group. But RNA comes also in 4 flavors, but one different flavor than DNA. So here's our RNA nucleotide, and instead of thymine, RNA is going to have a base called uracil, or U, which we're going to do in pink here. RNA also comes in 4 flavors. 3 of the flavors are the same as that of DNA, A, C, and G, but it has one unique flavor, uracil, in RNA as compared to DNA, which has thymine. Okay, DNA and RNA also both have a directionality. We call this end of the molecule the 3' end of the molecule, and this end the 5' end of the molecule. And the reason for that is it's named off of the sugar groups. This is called the 1' sugar. This is the 2' sugar. This is the 3' sugar. This is the 4' sugar. And then this is the 5' sugar. So this is the 3' end, and this is the 5' end. And the same is true of RNA. It has a 3' end and a 5' end. Now, this is important when we start to make a polymer of these molecules. So you guys are going to be upset about this, and I'm sorry, but I'm going to take my DNA and I'm just going to very quickly make a chain of DNA here attached together and put it like this, and then I'm going to copy this whole thing, paste it over here, and then go like this. So this is now a strand of DNA. This is the 5' end of this side and the 3' end of this side, and this is the 3' end of this side and the 5' end of this side. So we call these 2 strands anti-parallel because this one is running 3 to 5 in this direction, and this one is running 3 to 5 in this direction. I remember that Maggie was unhappy with this name, but this is the name we have. The DNA strands are anti-parallel to one another. Okay, if I were to go through and just mark up one side of this molecule of DNA with a sequence, let's say this was T, and then so if I were to go through here and say this side is A, C, A, T, G, you could then right away infer the other side of the molecule. It would have to be T, G, T, A, and C. And this is because we have this complementary base pair. A always binds to T, and C always binds to G. And the way that they bind is A binds to T with 2 hydrogen bonds, and C binds to G with 3 hydrogen bonds. So these are the only hydrogen bonds that are holding the two sides together. And as you know, hydrogen bonds and the rest of the bonds in these two strands, these two sides of the double helix, are held together by covalent bonds. So these bonds are the weakest bonds in the entire structure. And so it's actually relatively easy to unzip the DNA, which is actually going to happen, very importantly, during DNA replication, when one strand of DNA unzips and becomes 2 new identical strands. And we'll talk about how that happens much later on in the spring. Okay, so that is DNA. And how does this compare to RNA? Well, RNA would just be one side so an RNA molecule is going to look something like this. All these would have ends here, with the exception of wherever there is a thymine in RNA, it's going to be replaced with a uracil here. Okay, I think that's it for this screencast. I'm sorry about all my copying and pasting. Well, I guess I should also say that RNA molecules have a direction. This would be the 5' end of this RNA, and this would be the 3' end of this RNA. And yeah, major difference between RNA and DNA is that RNA is almost always found as a single strand, and DNA is double-stranded. Okay, that's all. See you in class tomorrow.